Hi class, this is Mr. Brenton here. These set of notes are going to explain how we set up controlled experiments. Basically, this is the way scientists usually do experimentation, and that's based on comparing one group to another. And we have names for those groups as well. So what we do is, this is important, that we compare the results to a control group to one or more experimental groups, and we're going to talk about what that control group is and why we need that control group for a controlled experiment. Well, the control group is called the normal group, and it's under normal conditions, and sometimes we say that it doesn't have the independent variable in it. It doesn't have the variable in it, so that you're leaving one group alone, and that's your baseline. That's where you're going to compare your other groups to see if there is an actual change. Remember, if we didn't have the control group, there would be no way to tell that actually this variable actually made a difference. And we'll talk about these variables, and you've talked about them in middle school, the independent variable versus the dependent variable. Well, we wouldn't know if the independent variable made any difference if we didn't compare it back to the control group. So that's the kind of thing that we talk about. We also talk about that the difference between the experimental group and the control group, there's only one variable that we're testing. So there'll be one difference, and everything else is identical. And that gives us that base to compare it to as well. So control group, left under normal conditions, doesn't have the treatment, doesn't have the variable in it that's being tested, and another way of saying it, it doesn't have the independent variable as well. Alright, so let's move on here so we can get to that. That's control group. And the other group we need to worry about, the other group is the experimental group. And the difference between, like we said, the experimental group is that it does contain the treatment. It does have the variable. Okay, it's the identical to the control group, and except for that it has a variable in it that we're being tested, and that's the independent variable. And we really should put that down. Let's hopefully I can get my pen to work here. Is the independent variable we want to say is the variable. So really what we talk about when they ask you about what's the variable, they're really asking you what the independent variable. So it's the variable being tested. We'll get to that in just a second. And that's the idea that you need to understand with independent variable. We'll get to that later. But the experimental group has that independent variable in it. Okay, so let's get back to, let's see, just give me a minute, scroll down to this question down below. Just move this down here. Okay, so let's move this here. This is a typical Regents question. We can read it together. The drugs usually used to treat high blood pressure do not affect the blood vessels in the lungs. Bosantan, is a new drug being studied as a treatment for high blood pressure in the lungs. In an experiment, patients treated with bosantan showed improvement in the distance they could walk without fatigue within 12 weeks. And they're asking you to design an experiment to test the effectiveness of bosantan as a drug to treat high blood pressure in the lungs. In your answer, be sure to state the hypothesis in your experiment, state how the control group will be treated differently from the experimental group, and state two factors that must be kept the same in both experimental and control groups, and state the type of data that should be collected to determine if the hypothesis is supported. Well, let's take one bullet at a time. State the hypothesis for your experiment. Well, that one has to be based on bosantan, and what we could say is, if we have some studies done already, that we can certainly say that bosantan would increase the or I should say decrease the blood pressure. You could say that, but also you could say that bosantan would also increase the distance that patients could walk as well. So either one of those could be acceptable because they're all predictions based on that. How is the control group going to be treated differently from the experimental group? Well, remember, the control group does not have the variable in it, or the treatment. You can think of it this way as a treatment. So what is the treatment? Well, it's actually listed for you in the second sentence. Is bosantan is the new drug, and it is the treatment. So we would not expect the control group to have bosantan. Okay, so it doesn't have bosantan in it. Whereas the experimental groups, or the experimental group, would be the patients that did receive the bosantan. So they're receiving the treatment. So you see how you can answer that question, and it's listed for you right there in that second line. So I can try to get my pen. Bosantan is the treatment. So that's what's given to the experimental group. Okay, state 2 factors that must be kept the same in both the experimental group and the control group. We have to think about things that could actually affect this data. It could be the age of the patients. It could be the temperature of where they were walking. If you're going to have these patients walking and measuring their fatigue, or the distance that they could walk, you'd want to walk in the same temperature. Their diets would probably be that you need the diets to be the same. You could even say that you're going to keep track of whether they're male or female too. So all of those things could play a factor in how long someone could walk. So those are the kind of things that need to be kept the same. There's several others too. But anyway, let's take a look at the next bullet. State the type of data that should be collected to determine if your hypothesis is supported. Well, if you're going to be measuring fatigue rates, you're going to be measuring their distance that these patients could walk. If you were to measure blood pressure, that could be another factor that you would be able to collect to see if this drug was meeting the needs of the patients or making a difference. So the type of data could be, like I said, distance that they walked, or blood pressure, whatever your hypothesis was based on this experiment. Okay, so those are some controlled experiments. Let's take a look at the next one. Okay, so a couple more vocabulary terms that we should be writing in our notebook. Again, to understand controlled experiments, we got to understand these terms of independent variable versus dependent variable. So independent variable is the variable being tested. It is what the scientist is changing, and we could even say that as well. Okay, so let's write that in our notebook. The variable the scientist changes. Changes. That's why I try not to write and record at the same time. Okay, and dependent variable is the variable that is being affected by the independent variable. Has to be measurable. It's usually part of the data that you're collecting. So we add that there as well. So it is the data being collected. Okay. All right. So that's independent variable and dependent variable. Let's take a look at. Try to get down to an experiment that may have an independent variable, dependent variable. If I can scroll down, let's see if we can do that. Okay. So if we take a look at this one, a student wants to bake the biggest loaf of bread in the local baking contest. Each contestant must use the same amounts of flour, sugar, and yeast, but is allowed to vary the type of sugar. Yeast is a microorganism that carries out cellular respiration, which produces carbon dioxide, making the bread rise. The student designs the experiment using the setup below to determine which sugar source, glucose, sucrose, or fructose, will cause the yeast to produce the most carbon dioxide and therefore the biggest loaf of bread. Well, again, we're looking for independent variable and dependent variable. Let me scroll up just a little bit more so we can actually see some of these things. And you can also see, well, what is the scientist changing in this experiment? Well, they were allowed to vary the type of sugar. So they varied, scientists allowed, or contestants vary the type of sugar, and you might say that's the independent variable. Okay, what about the dependent variable? What's the data that they're going to be collecting? The yeast is a microorganism, carries out cellular respiration, produces carbon dioxide. So if we're going to do this experiment, it's going to be basically what is their data that they're collecting, and we can actually scroll down and see the picture of this again. Okay, and basically in this experiment they used a balloon to collect the amount of carbon dioxide, or the gas that's being produced by the yeast. And we want to say that the sugar affected the amount of gas, or at least we think it's going to. And we're going to collect data on that. So when we abbreviate this, we can even say the independent variable, or we sometimes abbreviate that IV. So independent variable was the sugar, in this case, and the dependent variable was the amount of CO2 produced by the yeast. Okay, so that's a couple examples of how you're going to have to interpret some scientific experiments based on understanding about independent variable, dependent variable, control, and experimental groups. So we're going to incorporate that in some of our labs in class, so make sure you bring your notes to class as well, and we'll see you tomorrow. Okay, see you.